Can you hear me now? My sound mixer bar is working now, which tells me that hopefully we have resolved that. So, one more time, welcome Principles of Biology students. My name is Hunter, and this is content review for Biology 212. Thank you so much for drawing my attention to that. I do need that help here and there. Uh, Fantastic. So, again, a couple of admin things. I'll be streaming 4 p.m. live on Twitch every Thursday, and recordings will immediately, well, within an hour, go to the Portland State Learning Center YouTube channel where you can watch it at 2x speed and make me sound like a chipmunk, get a good laugh out of that. And you are eligible for extra credit for filling out these worksheets and submitting them by 5 p.m. on Friday, the week that material is presented. I've included my contact info on here. Do feel free to use me as a resource if you have questions about study habits or applying to medical school or whatever is most supportive for you. The workshop leaders and learning assistants and folks at the learning center that were really supportive for me, and I like to pass that forward where every opportunity that I have. So with that, um, let's dive into some biology. All right. So how's the general morphology of the various organs of a plant correlated with the functions of those organs? So how does the form inform the function? For example, why are leaves generally thin and flat? What structural advantage is provided to stems that have their vascular tissue arranged in a ring near the periphery of the stem? What characteristics of roots and root growth dictate what the vascular tissue be more so centrally located? So let's take a look at this first question here. Why are leaves generally thin and flat? Well, we've got our leaf and stem, big surface area, right? This is the think of leaves and what we know their function is. We know that they contain lots of chlorophyll. These are the pigments that absorb sunlight, and we know that those are really important for um, photosynthesis. That being their main job, it's functionally advantageous for them to expose a lot of surface to that sunlight to absorb that and not use a lot of energy on not expend a lot of resources in places they don't need. So again, thin flat leaves have that very high surface area to volume ratio, and as a result, they maximize surface area to expose photosynthetic cells to sunlight. So we need to use more pictograms today just because we're feeling like it. Next up, what structural advantage is provided to stems that have their vascular tissue arranged in a ring near the periphery of the stem? So I'm going to draw a stem sort of in cross section, and then we have this vasculature. So this would be a transverse cross section. If your stem is oriented like my pen, we're cutting it in half in this way. So what's the advantage of having that vasculature arranged in a ring? So stems do what? They support the rest of the plant, right? So they have their strongest tissues organized around the periphery rather than centralized. Um, but on the other hand, roots tend to be much more flexible, and so they have their strongest tissues, the xylem and phloem specifically, um, more centralized. So this, if we got stem, versus root. And I'm going to go back over the vasculature here in red, just because. Um, so this arrangement makes sense, and which support the. Let's see. Yeah, these rigid structures support the vasculature. And so think of this in terms of like the amount of energy that it would require to bend a copper pipe versus a copper wire of the same mass. That wire is going to bend a lot more easily, but that pipe, because of how it's structurally arranged, is going to require a lot more force. So you can also think of this in terms of, um, you know, I don't actually really like this example, but it's given here, so I want to. You could take a piece of paper. You could demonstrate this phenomenon using a rolled up piece of paper. First, fold the paper lengthwise a couple of times, take another piece of paper and roll it into a tube the same diameter as the folded paper, and it'll have greater structural integrity. You could have a volunteer hold each piece, um, and then put coins or some other mass on them to see which one will bend under less weight. So that's a simple experiment that you could do, but comes down to the different roles that these tissues need to fill, stem being more supportive, roots being more flexible and able to move into those areas where the nutrients are. All right, so how do water's properties of adhesion and cohesion help maintain the flow of water in the xylem of a plant? So adhesion sticks to itself, opposite. Adhesion sticks to other things, cohesion sticks to itself. And for those of you who are in general chemistry or have taken it already, you will remember that water is a polar molecule with a generally electronegative oxygen bonded to two electropositive hydrogens. So this is a polar molecule that has differing levels of charge, so it will adhere to surfaces that are positively or negatively charged. It's one of the and it will stick to itself really, really well. So it's one of those unique properties of water that make it so important in Earth's ecosystems and so essential to life on Earth. So, how does this help maintain the flow of water in the xylem of a plant? Well, if we think of that vasculature that I was drawing earlier, except now we're looking at it in a uh, different cross section here, and we've got our water molecules. The water molecules are attracted to each other, but they are also attracted to the surface here. So, as negative pressure, let's see, I usually use a different app to draw, so I want to see. No, it's not going to let me do that. Attracted to each other, also attracted to the walls. As negative pressure is introduced, it pulls the water up the xylem. Each water molecule pulls on the next molecule in the column. And so both molecules will go up. Yeah, as a result, the entire water column rises. So H2O coheres to itself. When negative pressure is applied, rising H2O drags the next water molecule. With it, leading to rise in the whole water column. All right. If water flows from a region of more positive, that is higher water potential, to a region of more negative, that is lower water potential, how does the water potential in the root compare to that in the soil outside of the root? So this is a nice reminder built into the question of what it means to have more positive or negative water potential. And this is true for most gradients that we'll talk about when looking at semi-permeable membranes. Um, things are generally moving from areas of high concentration to low concentration. So if you have a high water potential, it will want to move into an area with lower water potential. So if water flows from a region of more to less water potential, how does the water potential in the root compare to that in the soil outside of the root? Um, the water potential in the soil must be higher uh, than it is in the root. And we have a symbol for this, and that is psi. Water potential of the soil must be higher than water potential of the root. Another way to write this would be water potential of soil is greater than water potential of root. Okay. How does the water potential in the air compare to that in the leaf of a plant undergoing transpiration? To answer this, you have to know what transpiration means. And transpiration is basically water moving from the leaf into the environment, into the air. So this is a evaporative process, I believe. And so if transpiration is occurring, the air must have a lower water potential than the leaf. So water potential air is less than water potential of the leaf. Transpiration will occur. So taking this a little bit further, reading beyond the text here, the water potential of the air on a hot, dry day is going to be much, much lower than on a warm, damp, rainy day, right? Where your humidity levels are really, really high. Transpiration will not occur at the same rate. Or will occur at a decreased rate because that water potential of the air is higher than the dry air. All right. So a student uses a U-tube for a series of experiments. Sides A and B of the tube are separated by a membrane that is permeable to water, but not to sugar or starch. What result would be expected under the experimental conditions given below? And explain your answers in terms of osmotic potential, water potential, and the following equation. And this equation is the equation for water potential again. So I don't, yeah. Psi is water potential. Psi sub P is pressure potential, and psi sub S is solute potential. So as a hint here, I'm going to go ahead and highlight this because it's important. Solute pressure is always negative in this equation, and a 0.1 molar, that is moles per liter, solution of any substance has a solute potential of negative 0.23. Therefore, a 0.2 molar solution would have a solute potential of negative 0.23 times 2, since it's 0.2 molar, and that would equal negative 0.46 solute potential. Yeah. All right. So with that preamble, no, how do I yeet this person from the chat? Never to return. There. Okay. Sorry for the distraction. Experiment A, a solution of 10 grams of sucrose in 1,000 grams of water, and the molecular weight of sucrose is 342, that would be grams per mole, is added to side A, and an equal volume of pure water is added to side B. What will happen to the concentrations of water and sugar on the two sides over time? And explain. If you need help visualizing this, let's go ahead and draw our YouTube. We have a semi-permeable membrane in between. We have side A and side B. On side A, we have this 1,000 grams, which water has a density of 1 gram per milliliter, so we could just as easily say 1,000 milliliters of water, or 1 liter, for those who can do that division in their head, uh, and an equal volume of pure water. So we've got 10 grams of sucrose on side A and pure water on the other side. What will happen to the concentrations of water and sugar in the two sides over time? Process of osmosis, water wants to move from areas of high concentration to low concentration. Because side A has more solutes in it, right? It has all of this, was it starch? It has sucrose. Because it has sucrose in it, the the water behaves as though it wants the relative concentration of water to solute to be the same on both sides of this membrane. And so water is going to move from side B to side A where those solutes are. Let me make sure I'm not missing anything important here. They do want us to show the math on this, so I'm going to do that. All right. So if we have 10 grams of sucrose, let's do a little dimensional analysis. We know that sucrose has a molecular mass of 342 grams per 1 mole of sucrose. Grab my calculator. 10 divided by 342 gives us 0.029 moles. Is that right? Yep. We'll round that out to 3 moles of sucrose. And we have 1,000 milliliters of water. We also know from earlier in this, for every 0.1 moles of solution, we have a solute potential of negative 0.23. Yeah, 0.1 mole. Yep. And we have 0.03 moles per liter. This will give us negative 0.23. Let me think for a second. Yes. So negative 0.23 divided by 0.1 molar times 0.03 moles per liter gives us a final solute potential of 0.069. Now, going back to our original water potential formula, we also had to think about pressure potential. Just making sure you can see this behind my noggin. Yes. So our solute potential is accounted for do we have to worry about pressure potential? The short answer here is no, because we have a solution in pure water on side A with a equal quantity of pure water on side B. There's no difference in pressure potential between them. We don't have to worry about that. So our formula would just be plus 0. And so 0.069 is also our water potential. Okay, so that is the mathematical justification for what we would observe, which is water moving from column B into column A where all of that sucrose is dissolved. So in experiment B, we've got a solution of 10 grams of soluble starch in 1,000 grams of water. So very similar setup so far, added to side A. Assume the molecular weight of soluble starch is about 63,000 compared to 342. So the molecular weight is much, much greater. An equal volume of pure water is added to side B. What will happen to the concentrations of water and starch on the two sides over time, and explain how this compares with the results in experiment A. So I'm not going to redraw this because it would look very, very similar, but we can go ahead and run our same dimensional analysis. We've got our 10 grams of starch this time at 63,000 grams per mole. 10 divided by 63K would give us 1.58 times 10 to the negative 4 moles of starch per 1,000 milliliters of H2O. So far, so good. And our solute potential, therefore, equals yeah, we'll round this to 1.6 in our actual formula. So it's negative 0.23 per 0.1 molar. We've got 1 point, we'll round this, 1.6 times 10 to the negative 4 molar equals, so times negative 0.23 divided by 0.1 gives us does that check out? Yes, approximately. No. Yeah, it's fine. I'm being more precise than I need to be. So the answer key rounds this out to 0.0004 as our solute potential. And again, because we don't have to worry about our pressure potential because the amount of water is even on either side, we don't have to worry about that. So that is our final answer. So what would we expect to see in this case? So our water potential is much, much, much lower, but it is still negative on the side with the dissolved starch. So we would still see some movement of water to that column A where the solute is, but it would be much slower than the, what was it, sugar? Than the sucrose that was dissolved in the first experiment. Cool. Fertilizer generally contains nitrogen and phosphorus compounds required by plants. The nitrogen is often in the form of nitrates, and the phosphorus is in the form of phosphates. Based on what you know about chemistry and water potential, why would over-fertilizing lead to the death of plants? Adding nitrates and phosphates to the soil, if we stick with sort of the theme that we're talking about in this worksheet, um, you're decreasing the water potential in the soil, right? You're adding more solutes, water likes to hang out there. If the water potential in the soil is equal to or even less than the water potential in the root, then water's not going to flow into that root where it needs to be to nourish the rest of that plant. It can actually leave the root if water potential is lower than. Um, so if the water potential, well, let's see. The increased solutes, the nitrates and the phosphates, in fertilized soil results in a decreased water potential of the soil. If water potential of the soil is less than or equal to water potential of the roots, the plant won't take up water, which results in dehydration of the plant. One of the most common ways of killing a plant is overwatering. Why does overwatering kill a plant? Now, if in the previous problem the water potential of the soil was insufficient, can we solve this by just throwing more water into the soil? Is another way of asking this question. Um, and the answer is not necessarily, right? So roots, like any other living structure in a plant, require oxygen for cellular respiration in addition to that water, right? If you overwater a plant, you're filling all of the air spaces in the soil with water. The root tissue cannot survive without oxygen. So I want to write this and then we can talk about it a little bit more. Roots need molecular oxygen for cellular respiration. Overwatering results in no airspace. Overwatering equals no airspace for the oxygen to occupy, which results in cell death. Um, well, okay, actually, they bring this up in the next question. Fantastic. If overwatering kills a plant, then why can you sprout roots from cuttings of stems in water? And the difference here really comes down to the availability of molecular oxygen and its ability to diffuse in free surfaces of water, right? Like in a pond or in a cup of water, like you would sprout that branch in, that cutting in, versus water that is beneath the surface of the soil where it's not getting contact with that free air, right? So O2 from the air can diffuse, can diffuse into the water in a jug or a vase. So as a result, that cell membrane is going to get the oxygen that it needs to perform cellular respiration, right? Um, However, air spaces in soil are so narrow, right, and penetrate deep enough into the soil that oxygen cannot diffuse. And if you replace all of that air, it can't diffuse as easily. And if you replace all of that airspace with water, then the roots can't breathe at all. Xylem cells are dead when functional. Why must phloem cells be alive when functional? Xylem and phloem. Xylem, remember, is very structural, right? Xylem is used by humans for a great deal of our shelter, especially here in the Pacific Northwest, where we have a lot of timber, right? All of the wood that you would use to build a desk or something is xylem, um, is how I remember it, right? Phloem is involved with the flow of nutrients. Phloem is moving nutrients against their concentration gradients. It's moving, it's capable of loading sugars against a concentration gradient. And to do this, they must have semi-permeable membranes and must expend ATP. So as a result, they must be alive. So phloem, let's see, phloem moves sugars against concentration gradients. Process requires energy, requires ATP. Therefore, it must be living. What forces bring about xylem conduction, and how are these forces generated in the plant? So xylem conduction versus phloem conduction. So xylem, as we said, is dead, right? It's a structural part of the plant. The major force is the negative partial pressure generated in the xylem. Negative partial pressure generated in xylem, like xylophone. This force is generated through the transpirational loss of water in leaf surfaces. So earlier we were talking about those leaves that are exposed to the sun at the top of the plant, receiving that sunlight and that transpirational loss of water, evaporation happening at the top. Water moves into the air, decreases the water potential at the top of the plant, and water through adhesion to the vasculature of the plant in the xylem and cohesion to itself pulls water up through the column. So transpirational water loss. At leaf surface. Okay. What about phloem conduction? We'll keep this in blue just to keep it. The major force here is positive hydrostatic pressure in cells. Hydrostatic pressure in cells. So this force is generated by the difference in water potential in phloem versus xylem. Sucrose is actively transported into phloem to levels as high as 25%. And this potential difference pulls water out of the xylem and into the phloem, generating the hydrostatic pressure that moves substances into the phloem. So generated by water potential difference between xylem and phloem. Active transport of sucrose into cells results in a lower, lower water potential inside the cell of the phloem, pulls water. Also pulls water from xylem into phloem. All right. Refer to your diagram of the cross-sectional structure of a typical angiosperm leaf from activity 34.1 and correlate this structure, that is the type and placement of cells, with the activities of the leaf as they relate to photosynthesis, water conservation, and food and water transport. I do not have this diagram handy, but we can discuss it. So when the stomata are open in the leaf, carbon dioxide can enter and oxygen and water vapor can exit. I can draw a leaf. So we've got stomata, which have these, actually, I'm going to make this very oversized. Stomata have these guard cells. Water vapor and oxygen can diffuse out, and CO2 can diffuse in. It's the water loss through the stomata that generate the transpirational pull on the xylem. Right? How do I want to? Closing the stomata reduces water loss. However, it also reduces the amount of carbon dioxide available to the plant. C3 plants have a solution for this. Xylem vessels and phloem sieve cells lie in close contact, and these facilitate the movement of water from xylem to phloem to produce the hydrostatic pressure. Ooh, come on now. How do I get my pen to work again? Dang it. Well, that's okay. Maybe I can't draw anymore. It's frustrating. Really? Hmm. All right. Well, we'll do our best. Xylem and phloem are in close proximity. All right, scientists have measured the circumference of trees at 2 a.m. versus 2 p.m. And if they collect measurements when the ground has adequate moisture and the days are sunny and dry, they find that the circumference and therefore the diameter of the tree trunk is smaller at 2 p.m. than at 2 a.m. From your knowledge of the mechanics of water transport, suggest the reasons for this decrease in circumference. Will it let me type? Yes, excellent. Okay, we're back in business. So I kind of mentioned this earlier. On bright sunny days, the transpirational pull generated by evaporation from the leaves can be so great that it partially collapses or pulls in the walls um, of the xylem vessels. This is analogous to what happens when you drink a juice box, right? If you pull so hard on it, the suction on the straw that you collapse the box inward, um, you can suck on it so hard that the walls collapse. In the tree, this pull can be measured as an actual decrease in circumference. So when transpiration transpirational pull generated by evaporation is great. Xylem. Outline an experiment that would allow you to determine A, how fast a substance is carried in the xylem, B, in what direction the substance flows in the xylem, and C, what percentage of solutes are in the xylem at various distances away from leaves or roots. There are lots of ways to go about this experiment. We're going to talk about one in particular here. So xylem is dead when functional, which gives you a lot of liberty in terms of what you can do as far as inserting taps and probes and things like that. So, for example, you could place taps or measuring devices in the xylem along the side of the branch and place them at regular intervals in a straight line from near a leaf end to the opposite end of a branch. Okay. I wish I could draw this. It'd be so much simpler. Yeah, I'm aware that I'm in typing mode. I was working on a touch screen, and so I think I may have negligently tapped something, and now it's locked, so it's not letting me do that, which is fine. We can work around it. Um, anyway, take your branch, probes along its entire length, and in this now classical experiment, a guy named Bruno Huber inserted temperature recording devices at intervals along this tree branch, and he heated the water in the xylem at various locations along the branch and then recorded how, if at all, temperature of the water changed at the other recording devices. And this allowed him to determine what the water in the xylem, that the water in the xylem moved vertically. And by measuring the time it took for the heated water to move from one recorder to the next, he could also determine the speed of transport in the xylem. So let's do measure direction of flow. Probes at regular intervals. Heat water in the xylem. Record changes, if any, at each probe and at what time. That allows you to determine the direction and the rate of change or of flow. If you wanted to determine the amount of solutes present in xylem at various distances away from the roots or leaves, you could put taps and open the taps periodically and remove some of the fluid of the xylem, and then analyze the fluid from the various taps for its contents. So, for example, for the percentages of various minerals. All right. When researchers have tried to tap into phloem cells during experiments, they find that the disrupted phloem immediately stops functioning. However, aphids, the little bugs, can pierce through plant tissues with their mouth parts and locate individual phloem cells. Once inside a phloem cell, the aphids are essentially force-fed phloem sap. If the aphid body is removed from the mouth parts, phloem sap will continue to flow and can be collected. So if you place two aphids on a plant you are using for radioactive trace, let's see, prompt. You place two aphids on a plant you are using for radioactive tracer experiments. One is located 5 centimeters above leaf A, and the other is located 5 centimeters below leaf A. So we've got leaf A. Boy, it's not going to let me type on here either. One at the top, one at the bottom. You remove the aphid bodies so you can sample phloem sap from the mouth parts. You cover leaf A with a plastic bag and inject carbon-14 O2, a radioactive carbon dioxide, into the plastic bag. Leaf A is allowed to conduct photosynthesis for 10 minutes, during which time carbon-14 O2 is continually supplied to the bag. You start collecting honeydew from the two aphids' mouth parts from the time the carbon-14 O2 is first injected into the bag and every 30 seconds thereafter for a period of 1 hour. If you analyze the phloem samples for carbon-14, where would you expect to find it? And explain your reasoning. So the radioactive CO2 would be used by the cells in the leaf to make sugars for in photosynthesis. Can I type? Yes. So where would we expect to find these? These sugars would be loaded into the phloem. Because flow of sugars in phloem is from the source, an area of higher concentration, to sinks, carbon sinks, that is areas of lower concentration, you would expect to find some carbon-14 in the phloem samples sampled from the aphid mouthparts below the leaf. So, how do I want to write this? You'd expect to find some of the carbon-14 in the phloem sampled from the aphid mouthpart below the leaf. And so this happens because the sugars produced by the leaves are needed to feed the non-photosynthetic parts of the plant, such as the roots. Would the apical meristem also be a sugar sink? Yes, you would also see some carbon-14 in the samples from the aphid mouthparts above the leaf. Yes, you are absolutely correct. The growing bud at the apical meristem is also a sink requiring sugars from photosynthetic parts of the plant. Whoa, what just happened? I was on the wrong screen. Yep, 100%. All right. We are at 4:52, which is about the end of our hour, and that is the last exercise I had for you. So if you have any questions, feel free to drop them in the chat. I'll hang out for a couple of minutes. Otherwise, uh, go forth, reclaim, reclaim those 8 minutes. Enjoy the rest of your evening. Stay warm and dry. I hear we have more freezing rain coming tonight, or at least that was the forecast before I signed off uh, for the live stream. So do be safe out there tonight and tomorrow. And hope to see you next week. If you're also taking Gen Chem, I'm also doing a live stream for general chemistry students. That's 222 um, Fridays at this time, Fridays at 4 p.m. Unfortunately, I don't believe those students are eligible for extra credit, but if it is supportive for you, please do attend. Listen in the background while you're doing something else. You are so welcome. Thanks for attending live. Appreciate the little bit of human contact that we get, and thank you for catching the microphone. That would have been a real bummer.